Hello, my name is Heather Braden, and for my senior seminar presentation, I will present the effect of endogenous Basigin variant 1 on IL-6 expression for mouse monocyte immune response. And this research was performed with my mentor, Dr. Ockreiter. So the role of monocytes in the immune response cascade is very important for the body. Monocytes have an essential role in the initiation and resolution of the inflammatory response. These monocytes have an affinity to bind to surface receptor CD14 to secrete interleukins, a cytokine protein, and cascade the immune response. This cytokine protein interleukin signals to other cells, other neighboring cells, about bacterial infection, so that they can also perform the immune response cascade. The production of interleukin-6 proteins signal and induce a wide range of acute phase proteins. This IL-6 signal, along with other cytokines, enhances the TLR-induced inflammatory response from other cells. The secretion of IL-6 is into the blood for intracellular communication. Lipopolysaccharides, also known as LPS, are commonly found on the gram-negative bacteria, and these are detected by the toll-like receptor 4, also known as TLR4 complex on monocytes, and this induces an immune response. As you can see with this figure on the right, TLR4 does bind to the lipopolysaccharide layer on bacteria. And with this figure on the left, you can see that monocytes do produce IL-6 to direct to inflammation necrosis within the body. So the structure and function of Basigin, also known as emprin or CD147, there are two different versions of this protein. Basigin variant 1 is a transmembrane protein that contains immunoglobulin domains Ig0, Ig1, and Ig2, as you can see on the left of this figure right here. These are retina-specific proteins, whereas the commonly found protein, also known as general or Basigin variant 2, only contain Ig1 and Ig2 domains. Basigin variant 1 has 3 Ig domains to aid in lactate transport for cell-to-cell signaling to help them align with each other. This deletion or mutation of the CD147 gene results in blindness in mice caused by the lack of lactate transport. This is essential for the development of vision and can even have an effect on the development of cancer. The Ig1 and Ig2 domains were structured with a single disulfide bond by 2 cysteines, whereas the Ig0 domain has 3 cysteines, and this leaves 1 cysteine free. The study performed by Redzik and his colleagues suggests that the receptors present in the retina-specific Basigin variant 1 Ig0 domain are different than the Ig1 and Ig2 domains, and this is likely due to that free cysteine that is present. The free cysteine and dimerization in Ig0 were performed were studied to be the large contributor in IL-6 secretion. Using these quantifiable biological assays, it was discovered that the Ig0 domain present in Basigin 1 was a key simulator in the production and secretion of IL-6, and this leads to the immune response from the monocytes. This is supported due to the increase of the secretion of IL-6 in response to all 3 domains present, Ig0, Ig1, and Ig2, rather than just Ig0 or just Ig1 and Ig2. And this is due to a synergistic effect that all three domains have on monocytes. So for this experiment, Dr. Ockreiter and I wanted to determine if it was possible to recreate these results from Redzik's study using naturally expressed Basigin variant 1. We used an ELISA to determine the amount of IL-6 secretion into the medium as a result of these protein lysate treatments. So essentially, the amount of IL-6 secreted into the medium would have an effect on the absorbance values found using the spectrophotometer. So for the positive control, we used LPS, since the monocytes are able to detect the lipopolysaccharide layer on bacteria. LPS was a good positive control for this. The negative control was treated with PBS, as there would be no response, no IL-6 secretion as a response to this. For the experimental groups, we used 500 micrograms per milliliter, 250 micrograms per milliliter, and 100 micrograms per milliliter. And for this ELISA, we used a diluted BSA and PBS, primary antibody anti-IL-6, and secondary antibody APR. The PNPP substrate was used to determine the amount of IL-6 secretion due to the binding to the alkaline phosphatase product and tinting the solution yellow within the well, and thus the absorbance was able to be read using spectrophotometer at 405 nanometers. For the results, we discovered that, so for the data analysis, we averaged the absorbance values for 2 trials, and this was dependent on the protein treatment received. The absorbance levels determine the amount of IL-6 secreted by each group as a response from the protein lysate dosage. Standard deviations were also calculated using a single factor ANOVA to analyze the data, and these standard deviations were expressed using these error bars. The negative control was expressed using PBS, and positive control was expressed using LPS. There was no significant difference in IL-6 expression between the protein treatments with a p-value of 0.33, which is higher than the alpha value of 0.05. As you can see, both 250 micrograms per milliliter and 100 micrograms per milliliter had no significant difference between these experimental groups and the negative control of PBS, whereas the 500 micrograms per milliliter had a large margin of error, and actually the error bar overlapped both PBS and LPS controls. The LPS did have a significant increase between this group and the negative control and the 250 and 100 microgram per milliliter experimental groups. So the results display that there would not be a significant difference between experimental groups and the negative control. So this means that according to the data collected, endogenous Basigin variant 1 did not induce IL-6 expression, and our results were different from the results that Redzik received in his study. As mentioned before, the group that received the 500 micrograms per milliliter protein lysate treatment had a large margin of error and overlapped both controls. And this could indicate that more trials would be needed to produce a smaller standard deviation and determine if Basigin variant 1 had an impact on IL-6 secretion. This study is extremely important to research due to the stimulation and regulation of matrix metalloproteinases, also known as MMP, and this is regulated by Basigin, also known as CD147. Now, these MMP enzymes are responsible for breaking down the extracellular matrix of cancer cells and are a contributor to the spread of cancer throughout the body. And thus, understanding Basigin is essential for cancer research and how to stop the spread of cancer within the body. And here are my references. Thank you so much for listening to my presentation.